And then she moved to the United States and received a PhD degree in chemistry from Caltech in 1973. And after the postdoctoral training, and she then joined the Cold Spring Harbor Laboratory. And so during that time, she used adenovirus as the model system, and her works led to the EN discovery of the RNA splicing and the alternative RNA processing. So that was a major contribution to the field of RNA biology. And in 1984, she then moved to University of Rochester. And in 1993, she moved again to the University of Alabama at Birmingham. And she is now the professor in the Department of Biochemistry and Molecular Genetics. So Professor Cho right now works on the human papilloma virus, HPV, and she has revealed the molecular mechanism for the HPV DNA replication and the viral pathogenesis using elegant biochemical and fluorescent microscopic techniques. And due to her excellent research, she has received many honors, including the foreign associate member of the National Academy of Sciences in 2012, the fellow of the American Academy of Microbiology in 2013, and the academician in the 2014. So we are glad to have this opportunity to listen to her talk. And the title of the talk is Model System to Investigate Molecular Genetics of HPV. So let's welcome Professor Cho.
Thank you for your generous introduction. I'm very much honored to be a speaker in this symposium. It's a very tough act to follow. All the previous speakers, everybody's so outstanding. I just hope I don't bore you. Okay. Let's see. Let me first outline what I'm going to speak about. It's about the infection and the disease it causes, and then a little bit about the viral genome and its expression, and the natural history. Then a few slides about mechanism of HPV oncogenesis, and then actually much of the talk will be based on the model systems. And we study the genetics and also try to identify antiviral inhibitors. So this is a small DNA virus, double-stranded DNA, only 7.9 kilobase pairs long. And it infects lots of vertebrate, but they have very stringent species and tissue tropism. So human papillomavirus will not infect animals, and the animal viruses will not infect human. And the human papillomavirus is virtually ubiquitous. Everyone in the room has them. It's just a different body size. Some of them are cutaneous specific, some are mucosal trophic. And the infection, what is the outcome? It's just epithelial hyperproliferation, so you have a wart, okay? And the wart sometimes called papilloma, and sometimes called condyloma. Then we usually have very good immune systems, so the wart will persist for maybe half a year to a year and a half, and then usually goes away. And they may go away permanently, or they may persist, go into latency to be subclinical. But they can be reactivated during stress, during immune suppression, or repeated wound healing. So this is a very successful persistent virus. Now, what are the diseases actually you see? Then you have warts, which could be on your hand, on your leg, or on your palm of your hand or bottom of your sole. And the most concern is the mucosal trophic viruses. And they can infect anogenital tract, and these infections are usually sexually transmitted. That's why most of the study are on the mucosal trophic viruses. And they cause genital warts and condyloma. And then some of them can progress to high-grade dysplasia and cancer. And that's why most of the efforts are on this group of viruses. And some of the mucosal viruses also, in fact, on oral cavity, most prominently causes juvenile adult onset laryngeal papilloma. These viruses sometimes can, the infected cells can grow down the trachea into the lung, and that usually causes death. Even though they are warts, they're not cancer. So now there's a group of human papillomavirus called high-risk virus. Why are they high risk? Because they can progress to cancer. And there's about 2 dozen such viruses, most prominently type 16 and 18. But progression to cancer usually takes many, many years. So early diagnosis and treatment is the only way to prevent cancer. But now we do have something better. Let me continue this subject. So all the cervical cancers are caused by human papillomaviruses, this high-risk virus. And most of the vaginal and vulvar cancer as well. Then penile and anal cancer, most of them are caused by papillomavirus. And even 25% or higher in different studies, up to 50%, head and neck cancer attributed to the high-risk virus, mostly HPV-16. Then globally, it's estimated that about 500,000 people will die of HPV-caused cancer, and mostly in resource-poor countries which have no early diagnosis or even any treatment. So what about prevention? And there is a prophylactic vaccine, but this type is restricted. So if you have a vaccine against 16 or 18, you can prevent 16 and 18. You don't prevent 33, 35, or so on. But remember that maybe 2 dozen of them can cause cancer. Now the vaccines up to 9 types. 7 are oncogenic or high risk, 2 are non-oncogenic, which is 6 and 11. Those are the ones cause genital warts, mostly genital warts and the laryngeal papilloma. The high-risk viruses, 16, 18, are included in the 9mer. But on the market is the tetravalent vaccine, and it's Merck is selling. The 9mer has been approved, but we have not seen it advertised for distribution yet. So, and the main problem is that there are no pharmaceutical drugs that's reliably effective, and that's what's really lacking. And what we need is such inhibitors, because once the virus is there, if a person is infected and the prophylactic vaccine will not do anything, you need treatment. But there's none so far. So let me just say a few words about this virus. Genome organization. This is a linear map of the circular genome. Okay, all the transcription goes one direction. The very few genes. So the early genes were mainly devoted to maintaining and the viral DNA and amplification. Late genes for capsid protein. And this is a non-coding region called upstream regulatory region. It contains DNA replication, origin, as well as transcription promoter enhancer. And there are 2 genes, E67 plus E5 here. These 3 are considered viral oncoproteins. And E67 is target tumor suppressor proteins to recondition the cell to support viral DNA amplification. And E5 is a transmembrane protein, enhanced tyrosine receptor kinase, receptor tyrosine kinase signal transduction. And the virus encodes E1, E2 protein. Those are the two proteins responsible for maintenance as well as amplification. And there's also a E4 protein bound to cytokeratin. And we don't know what its function is, probably to facilitate virus release. And importantly, E5, E6, E7, they're all downregulate host immune surveillance to establish a persistent infection. Then the productive virus infection is a very difficult subject to approach. The virus cannot be propagated in conventional cell culture system. Okay. And so from extensive research, and now we know the virus establishes infection only in the basal cells of epithelium, okay, and through a cut. So just having virus on the surface of your skin, if the skin is not broken, you're not going to get infected. Actually, it's very hard to get infected by HPV, except when you have wounds. The viral early promoters are activated in the undifferentiated keratinocyte upon gaining entry into the basal cell. But once the wound healing is over, the promoter is shut down. So the virus is maintained in very low copy number. High-level viral transcription DNA amplification only happens during the cell differentiation in the mid and upper strata. Then the capsid protein is only produced, detected in very few cells, in the superficial cells. And then the progeny virus is shed with the dead skin. So there was never a viremia and never causes cell death. So it just take the conveyor belt as a tissue differentiate, it produces virus. And that's why it's very hard for the immune system to detect it. It's never viremia. Now, this is a very old slide, but I like to show it. In those days, we are in situ hybridizations using ribo probes, tritiate label, ribo probes. And the reason I like to show it is because this patient, this side is normal. This side you see is clearly abnormal. This is cervix. This happened to be a HPV-16 in HIV positive patient. On this side, you can see the gene expression by in situ hybridization. The white dots, that's the autoradiograph. You can see that all differentiation dependent. This is the surface. This is the more mature cell. This is the basal layer. So you can see the normal tissues completely not infected, only the diseased tissue containing HPV. This is the proof, the gold standards that certain diseases are caused by the virus. And then we can also look at the viral DNA amplification. This is also a HIV positive patient. Viral DNA amplification also in the mid to upper strata, and the lower strata is complete. It can detect anything at all, and usually it's considered maybe 20 to maybe 100 copies per cell. Okay, and again, this is an autoradiogram, and you can see this side is less differentiated, higher grade dysplasia, and yet on the surface there are a few cells that differentiate and can support viral DNA amplification. And so how do you study such virus? Because when you grow cells in cell culture, they are proliferating, they're not differentiating. So we'll get to that in a bit. And this is a more recent in situ hybridization, laryngeal papilloma. This is a low-risk virus. You can see the fluorescence in situ. The green signals are the viral DNA. The signals are only sporadic, not in all the cells, but in the differentiated, some of differentiated cells. And typically, in HPV infected patient, very, very few cells makes L1 protein. Okay, these are immunohistochemistry, and very few cells, except perhaps in HIV patients who's immunosuppressed, then you have a bit more virus produced. So the issue is that in a normal tissue, the only cell that can divide, can support the cell division and replicates DNA is only the basal as well parabasal. Normally, the parabasal cells are the so-called transit amplifying cell. They divide day in every day. One of the daughter cells remain behind, and one daughter cell is pushed up to differentiate. Okay? And so we are here probing for a cellular protein called proliferating cell nuclear antigen, and that's essential for cellular DNA replication. Okay? And basal cells are actually fairly quiescent, and only the parabasal cells divide. And yet the viruses are dividing, replicating DNA up here, and yet we know the virus has only one enzyme, which is U1 nucleate, DNA helicase. So everything else has to come from the host. So that tells us the host DNA replication machinery must be turned down. So indeed, this is a benign ward, HPV 6 or 11. This is a genital ward. You can see now we're looking at PCNA, and lots of differentiated cells are positive. Yeah. We can still see the parabasal cells are positive, so is many differentiated cells. Basal cells are not really that positive. They still remain relatively quiescent. So a ward is basically hyperproliferation of the parabasal cells to generate extra tissue. And so since all the DNA replication machinery is turned on by the virus, we asked whether the host DNA is also replicating. And this is a laryngeal papilloma freshly excised from a child, and we raised it back from the lab, dipped it in tritiethymidine. This is again the old slide, so we're still using tritiethymidine. And you can see indeed the host DNA is replicating in the differentiated cells. So how does it do this? Okay, how does the virus do to replicate in cells that already exit the cell cycle, in the differentiated cells? So, and also why certain viruses will cause cancer. And this answer comes from the virus E7 oncoprotein and E6 protein reestablish in a condition conducive to support viral DNA amplification. And the targets were PRB and P53. And these are two major tumor suppressors that control cell cycle entry, cell cycle exit, differentiation, senescence, apoptosis. So you can see if these genes, instead of being activated in the differentiated cells, but activated in the undifferentiated basal cells and continuously activating there, then it could cause a problem, becomes a double-edged sword. So wrongly activated oncogene can cause problems. So how do we study this virus? And this is a system two of our students developed back in the early 90s. So we can take a tissue explant, most of the time, actually virtually all the time, we're using primary keratinocyte recovered from neonatal foreskin circumcision. And many labs using immortalized epithelial cells. So we can introduce viral gene in there using a retrovirus-mediated gene transfer. We can also transfect a DNA, and as we produce virus, we can also infect these cells with bacterial virus. So we grow these cells normally in petri dishes, and then usually if we have transduction or transfection, we have a selectable marker, we can select for those cells. And in the meantime, we make a dermal equivalent which is collagen embedded in it with fibroblasts. That is like our dermis layer in our skin. Then we put the keratinocytes on top, roll them for a couple of days, and then once it's reached confluence, we raise the entire assembly to a platform, onto a raft. That's why we call it raft culture. And now the whole assembly is exposed to air, and then within 10 days or more, we have stratified the epithelium. Okay? So it's a pretty long process, starting from recovering keratinocytes from foreskin. And if you look down the petri dishes, here's our raft. Here's the collagen with cells, with tissue. And this is when you embed the tissue, and formalin fix embedded the paraffin in the tissue section, and you see that this is just like regular skin, okay, with all the strata, even the square. So now the question is, does it really behave very well like real skin? Then we use a retrovirus, either empty or putting E6 G in there. We can see the PCNA in the basal cell, and BRDU, which is a thymidine analog. We no longer use the radioactivity, so we can detect BRDU incorporation in the host DNA use empty. So they are right where they're supposed to be, and nothing above. Then if we put E7 in there now, you can see now PCNA is virtually in all the cells, some cells more than other cells. And BRDU incorporation is just like the laryngeal papilloma I show you. It's not in every cell, but again, it's in the differentiated cells. And we can actually do mutagenesis in this system, mutate different parts of the E7. Turns out you need the part that binds the PRB pocket, and you need CK2 a motif and its substrate. So if you mutate any of the three, the E7 is dead. So now we know the E7 condition the cell to take it back to S phase. So all the DNA replication machinery is available for virus to use. Now, turns out it wasn't as simple as that. We found out the target is not really PRB in the differentiated cells. The target is a PRB-related protein called P130. And P130 is very high in the post-mitotic differentiated cells. And now BRDU now is in red, P130 in green. If you put E7, P130 is gone, BRDU is incorporating into the differentiated cells. You make a point mutation, E7 is no longer active, and then P130 back to in the differentiated cells, and BRDU in the basal cells. And this is true for HPV1. This is a plantar wall E7, and the laryngeal papilloma E7. These are less effective than the oncogenic HPV18. Again, if you make a mutation, then it's just like this one. This is also in the pocket protein binding domain. So you can do genetics, you can find out what's the target. If you look for PRB, you will not find PRB in these differentiated cells. So now, I told you E6 has no effect. So if you want to study other viral genes beyond E7, we must have to have a system to introduce the entire genome in there, in the background of differentiating epithelium. So everything has to be like in vivo. So how do we do this? And this is developed by another two students. So the many labs, remember the virus cannot be grown. So all we have available to us are cloned viral DNA. And many labs excise the viral DNA from the recombinant plasmid, just transfect into keratinocytes, either primary or immortalized cells. But those transfections are efficiency is very low. They usually don't get very far. So the wild type doesn't work very well. The mutant works maybe less, so you can't really tell what's going on. So we decided that we needed a new system to study and use the Cre-Lox system. This is, of course, used heavily in mouse genetics. And this is a bacteria, Recombinase. Cre is recombinase. It works on LOXP site, which is 34 base pairs long. If you open up the HPV genome at the right place and put the LOXP site there, co-transfect these two plasmid, those are supercoiled, and when you get inside, you get inside the cell, and Cre works on the LOXP. Now you have two plasmid. Okay. And now this remains supercoiled. It's a perfect substrate for DNA amplification, replication at transcription, RNA transcription. And then actually 30%, 20-30% of cell survive the selection. That means supercoiled DNA transfection is not bad. It's the linear DNA transfection that doesn't work. Now if we make the rat culture, wait for about 2 weeks, then formalin fix embedded the parafilm, did section and do in situ hybridization, fluorescent in situ. Now we see lots of DNA amplifying in the differentiated cells. Again, here's the basal layer. Okay. And we also did southern blood hybridization and can estimate about 5,000 copies of our DNA per cell. Of course, not all the cells are amplifying, so some of these cells have really very high copy number. And actually, if we look for L1, we can see across the entire rat culture in the superficial cells. This is squam. Okay, these are dead cells. This is live tissue. And you can see it's right at the border. That's where the L1 is being expressed. And then we make lots of virus. And the virus is infectious. We can infect primary keratinocytes and then reproduce the entire life cycle. And what the two students did is did a very thorough study of this system. What we find out is that not only E7 induced S phase entry, it also induced a prolonged G2 phase in these differentiated cells. And the E7 induced a DNA damage-like response. That is the reason leading to a G2 phase, okay, G2-like phase. And this is when the virus DNA amplified. This is quite a surprise. We all thought when the host DNA is replicating, the virus would just piggyback and taking the available machineries and substrate to amplify, but that's not the case. Viral DNA actually amplified during the G2 after the S phase is finished. So to us, it means that there are no spare parts once the cells are engaged in DNA replication. So the virus has to wait. And also, very interestingly, we found that as the virus DNA amplifies, the E7 activity actually goes down, eventually becomes extinguished and stops. Why? Why does it do that? We know this because the P130 comes back. Okay, I'm not going to show you the data. It's all published. When the cell E7 activity goes away, cell, of course, P130 comes back, cell exits the cell cycle, and the virus switches to the late phase. Now it's time to make the capsid protein to package the progeny virus. So now it all makes sense because we can do post-chase experiment and to confirm all these interpretations. And by looking at one spot, one time point in patient specimen, you cannot figure out all this. So now we have the productive system. It's time to study E6 function because E6 cannot be studied. If you make an E6 mutation, the plasma usually is gone. When by the time people are ready to do the experiment, very much down the line because they have to take a long time to select for transfected cells. But in our case, we can do the RAF culture within a week after transfection. So, and we were able to do the experiment. The E6 is a multifunctional protein. It targets many host protein for degradation in the case of the high-risk virus. And one prominent target is p53. Actually, all the small DNA tumor viruses target p53. So the question is, which one is E6 doing, targeting in this productive phase? So we, two other students made E6 mutant, different E6 mutants. Okay? Some are null mutations, some are point mutations. And the key thing is that we want to see if we cannot eliminate, cannot target p53, what would happen? Is p53 the target? So again, we can, this is a wild type. Lots of new viruses being made. L1 is a huge amount, but this mutant, hardly any. Okay, we can keep it all the way out to 22 days. You will not see capsid protein, and actually very few DNA amplification. And what happened here? You can see if we have this day 10, day 12, day 14, day 16, you probe for p53. p53 is very abundant. Viral DNA, not very many. Okay. So that tells us p53 really is one that controls viral DNA amplification. Now, if you rescue this mutant using a retrovirus expressing E6 or using a retrovirus expressing p53 shRNA, and then you can largely restore viral DNA amplification. In the meantime, p53 goes away. So now we know p53 is the one that controls viral DNA amplification. And prior to this, there were many speculations. Maybe the cells will undergo apoptosis, and p53 is there to prevent apoptosis to give virus time to amplify, which turns out not true. We never see apoptosis in persistent when we even don't have any E6, E6 no mutant. We don't see apoptosis. So now let me switch gear, talk about pathway-specific inhibitors. HPV-induced cervical cancer, there are many mutations detected in several studies, but they vary from lesion to lesion. So each patient may have a different mutation. So it's hard to try to find inhibitors targeting a common target. So what our strategy is to figure out what the virus needs. If we can target what the virus needs, then we can control the virus production or disease. So that brings me to E1 protein. This is the DNA helicase, the only enzyme, and it's the most conserved protein. And it's the only enzyme. So why is it so interesting? And this is the origin, consists of E2 binding site and E1 binding site. E2 binds as a dimer, and it recruits E1, which binds a dihexamer. So everything else comes from the host. So that's why the cell has to go back into S phase. Now, this mechanism of replication is conserved among all HPV, even animal papillomavirus. So if we can target this system, then the drug should be effective for all the viruses, regardless which virus type or whether even animal papillomavirus. Much of the strategy in the literature using siRNA, which would be type specific, because the sequence of each virus is slightly different, or therapeutic vaccine targeting E7, again, that would be type specific. Here we're trying to find a drug that you don't have to type the virus, you can just hit it. And we were lucked out. And with the DNA virus to replicate the nucleus, and it was quite a surprise when two of our students discovered the virus shuttles between nucleus and cytoplasm. Okay, and the control region is all within 50 amino acids near the N-terminal portion. has nuclear localization sequence, nuclear export sequence, and there's a cyclin binding motif, and this is a bipartite nuclear localization sequence. This sequence is not really active until ERK, okay, which is a MAP kinase, ERK, phosphorylates these two serines. And the nuclear export sequence is pretty strong, dominant over nuclear localization sequence, unless it's inactivated by phosphorylation on this serine. Here's the nuclear export sequence. And the kinase is CDK, and that also tied in with the cell cycle issue. And interestingly, here are two motifs. That's a binding motif for the MAP for ERK and JUNK, okay, mostly ERK. So this is the model, E1 in the cytoplasm, default pattern. And when the cells in the right condition, MAC12 activates ERK1 or 2, which then phosphorylate the serines, and the E1 goes into the nucleus. However, the exportin dot here, which is dominant over the nuclear import, the E1 protein gets shut right out. Unless now we have cells in cell cycle, and this additional serine is phosphorylated by cyclin-dependent kinase, then the E1 stays in the nucleus to support viral DNA amplification. So here, just want to just show you one slide. This is GFP-E1, which is functional in transfected cells. We just mutate the cyclin binding motif. You can see the nuclear export sequence no longer can be inactivated, so they all comes out. So now this E1 will not be functioning. Okay, what is so interesting about this discovery is that the conservation of all these signals. Okay, every different, doesn't matter high risk, low risk, or cutaneous virus, they all have the same signal. And even animal virus, except for bovine papilloma virus, doesn't have the nuclear export sequence. And this is also true for the MAP kinase docking domain for all the viruses, including bovine papilloma virus, all conserved. So the hypothesis is that if we can target this shuttling, we should be able to control the virus. So that was all discovered with HPV11, which is a benign virus. And our rat culture is the high-risk virus, the E1. Okay, so the E1 helicase, we think should behave the same because it conserves regulatory region. So we made mutations, many mutations. I just want to show you one. That's the mutation no longer can inactivate the nuclear export. When that is compromised, the virus is gone. Okay. Here is E1, the S1 point mutation. This is the whole virus, okay? And you can see any sign of it. But if you mutate to S2 phosphomimetic mutation, you restore amplification. So this is just as we had hoped and anticipated. And the other way to, but that's just a mutation to show that the system probably will work. And the real test is that can we use MAC inhibitor to inhibit viral DNA amplification? Because now you can control its nuclear import. And here's untreated. This is BRDU in red. Green is viral DNA. On day 14, you can see lots of amplification. Now we treat with one of two MAC inhibitors, which is upstream from ERK. You can see that both will eliminate viral DNA amplification from when we treat them from day 10 to day 14. Just during the time virus is supposed to amplify, so we eliminate it. but the host DNA is still replicating. So that means the virus is not gone because we treated very late, and when the epithelium is already established. Now, if we treat them from very early on, day 2, when we lift the RAF culture, and this happened to be a RAF culture, has a very low copy number per cell to begin with. And on day 14, if it's untreated, we are probing for PCNA. Remember, PCNA is induced by E7. Okay, cell cycle is induced. Cell cycle reentry is accomplished by E7 expression. PCNA virtually in all the superficial cells. Okay, and this is uninfected cells, only a few cells in the basal parabasal cell. If we treat it for a long time with the MAC1/2 inhibitor, you see that many cells now no longer have PCNA. Okay? We suspect those cells no longer harbor virus, because once you have the virus, even one copy, it will induce PCNA. But we do. Some of them still do, but many of them don't. So that was pretty exciting to us. We have other inhibitors that actually have a different effect that causes a massive cell death. Okay? So there are many inhibitors we are testing. Now, in the last few minutes, I want just to tell you why there's low-risk virus, there's high-risk virus. And it turns out there's a big difference. They all do the same. They're all amplified in the differentiated cells and produce virus. The difference, major difference, the low-risk virus can degrade P130. That's what they need to do to amplify. The high-risk E7 not only degrades P130, also degrades PRB. This is a major tumor suppressor. Now, low-risk virus can inactivate P53 within, that's enough to support the viral DNA amplification. The high-risk not only inactivate, also degrades. In addition, also can degrade many PDZ domain containing proteins. These are the controlling cell polarity and signaling, so very important protein. Then the high-risk E7, E6 can also activate H3. This is the one maintaining the telomeres. So these basic differences really enable the high-risk virus, E6, E7. If you constitutively express these two oncogenes, you can immortalize primary keratinocytes from foreskin or from cervix. So that's the major difference. Why there's a high risk and there's a low risk, nobody understands, but they exist. Now, this is a slide to show, again, a very nice HME in this patient. This side is low risk, low grade CIN1. It's productive infection. But as you just travel tiny bit distance away, you can see these cells, different morphology. This is a high grade lesion. Within very distance, you can see the gradation. Then we did in situ hybridization in one of the cervical cancer patients. This is not lay gene or viral gene. Other viral gene is all E6, E7. That's how much there is. If you recall earlier, there's hardly anything. There's very low amount in the undifferentiated cell. Here is all undifferentiated, but it's highly expressed. So that tells you this patient is in big trouble. I think she died within a few months after the biopsy was taken. Now I can summarize that there's alternative outcome for HPV infection. HPV infect the basal G0 cells through a wound, and during healing stage, virus early gene is expressed, and when there's the immune deficiency or temporary depression, we have a differentiation dependent productive phase. That's true for high-risk virus and low-risk virus. And then immune system intervention could cause regression to latent infection or clearance. So this can go back and forth if it's latent. But if we have repeated wound healing in the basal cells, then the basal cells could be transformed neoplastic progression, and this repeated activation due to wound healing of the basal cells, as well as immune suppression, we can lead to high-grade dysplasia. Eventually, we have epigenetic changes in the host gene. It can lead to immortalization, transformation, and invasive cancer. Now, the lab is also working on finding inhibitors for cervical cancer. Again, we're using pathway-specific inhibitors. We know, and other labs has reported, the PI3 kinase, AKT, those are activated in cervical cancer. So this is, I just want to show you one slide. This is a dual inhibitor of PI3 kinase. mTOR is also activated by HPV infection. So this is a normal keratinocyte. It's affected. It's thinner, so the cell proliferation, the basal cell level is affected, adversely affected, but there's no apoptosis. All this black, these are melanocytes. Okay, in the south we have a lot of black babies. This happened to be one of them. But this is a cancer cell line called CHA, harboring HPV-16 integrated genome, and when you treat it with this dual inhibitor, completely obliterate the cancer. Okay. But cancer cell lines, they've been established in the lab for many, many, many years. They may not resemble the original cancer anymore. So the lab now is switching to patient-derived tumor, developing xenolines in skid mice. Okay? And those skid mice tumors can be actually grown as what's called an organoid. Okay? And then we can treat them using our own hypothesis as to what might work, targeting the pathways the virus needs. Again, we're not screening lots of drugs, but we are doing it the smart way. Basically, I like to say the only a few drugs we need to test, because those are the common pathways the virus requires. So that's the work that's still going on. And preliminary data says it will work. Okay, so we just need to validate that our organoid is resembling the original tumor. Okay. And that's the I think everybody does patient-derived lesion have to prove that they are looking at the right thing. Okay, now let me just show you the. So we have model systems. And this is the oldest slide, and maybe the people that did the work. And actually a couple of them from Taiwan. Okay, and he's now in Taiwan, and Liu Jiehua and Wang Shukun is still with us. A lot of work done by Sanji Banerjee. And the rat culture system pre-labs was established by Wang Shukun and Duffy. And E6 work, E6 star is done by Yang and Wang. They are coupled. Okay, and this lab is done in Tom and my lab. We've been working together since we were married in the 70s. So that's it. Thank you.
Okay, the seminar is open for questions. Let me start with, I have again a naive question. So I'm curious, why is the E1 DNA helicase is required for the HPV DNA replication? But it used the host, the DNA polymerase for DNA replication. But DNA polymerase does not need a helicase.
Yes, we did. MCN.
Really?
Okay, I need the MCN.
Okay.
The MCN is the helicase. It's a sister unit.
Okay.
For the host. Viruses is on.
Okay. So have anyone tried to develop drug using this E1 helicase as the target?
Well, the helicase, this ATPase, but that's so common.
Okay.
Okay, and it's very hard to be so specific to target that one is ATPase without targeting others. So that's why we thought, if we control. And what's interesting here is that in our drug, I didn't have time to show, is that we can treat them for 2 to 3 weeks for this MAC1/2 inhibitor, no toxicity. And the reason is that the skin, the key MAC kinase is junk. If you use junk inhibitor, the tissue does not develop, virus doesn't amplify. But the MAC inhibitor, the host can switch to junk, but the virus cannot. The virus requires ERK. So that's why it's a nice target without a lot of adverse effects.
Okay, thank you.
For virus, it's also, you can treat them topically. You don't have to treat them systemically when they are still a ward. And that's the other beauty of this system. A lot of the drugs that have toxicity because they apply systemically, with injections or taking pills or whatever. But for the benign stage, before they integrate, then you can treat it topically.
Okay, thank you. Yes.
Hi. Oh, a nice talk. I have two curious questions. Since you said everyone been infected by HPV, okay, so for adult, if they want to take a vaccine, would the vaccine still useful to the adult? My question is that since the virus is going to latency, right? So would the vaccine possible to block the reactivation of the virus?
Because it's prophylactic. The vaccine is made of L1, VLP, virus-like particles. So only prevent new infection. The existing infection doesn't do anything. It's
okay, so you cannot block
L1.
So cannot block the reactivation.
No, because remember, the capsid protein is only made in the superficial dying cell. Okay. By then, it's too late. It can prevent new virus coming in, but it cannot stop virus that's already there. So it's only prophylactic. It will prevent new infection by that particular type of virus.
Okay. And also about the E1 protein. So E1 protein after phosphorylation, it will be retained in the nucleus. Will it because E1 protein after phosphorylation will form a tetramer, a hexamer, because it's a helicase, right?
It's a helicase.
Yeah, right. So a helicase. So phosphorylation may assist the formation of helicase. Therefore, become too bulky to go back to the cytosol.
Actually, we are not sure that is the reason. We know some people express E1 ectopically, so presumably fairly high level, maybe higher than the virus ever makes. It kills the cell. It's a very potent helicase when you express at high level. Maybe that's why you need to control only the right time when it's coupled to the viral DNA amplification. That right time to support viral DNA amplification. Then the E1 gets made and gets shipped into the nucleus.
Okay.
Yeah, so that's our interpretation.
So any more questions? Okay, if not, then let's thank Professor Cho once again for the wonderful